Good afternoon, everyone. I would like also thank organizers for kind invitation and giving me the opportunity to share more data with you during this wonderful conference. The subject of my today's speech is Pup ultrasound vocalization in the neurodevelopmental model of schizophrenia, and the main goal of the study was to analyze of vocalization of infants that were prenatally exposed to metalazoxymethanol. At the beginning of my talk, I will introduce you to schizophrenia in humans, and I will focus on negative symptoms of this disease. Next, I will quickly remind you the information about how vocalization changes in animal model of schizophrenia. Then I will present my results, and I finish my talk with the conclusions. Schizophrenia is chronic and severe mental disorder that affects how persons think, feel, and behave. The symptoms of this disease, which can be very disabling, fall into three categories: positive symptoms, negative symptoms, and cognitive impairments. Positive symptoms are psychotic behavior, not generally in healthy people, such as hallucination, delusion, and disorganized thought, which also manifest often in disorganized speech. In turn, negative symptoms are associated with disruption to normal emotion and behavior, and cognitive impairments reflects patients' problem in concentration, planning ahead, decision-making, and organization of their life. Schizophrenia is usually diagnosed from adolescent to early adulthood when the first psychotic symptoms appears. Although not invariant, negative symptoms are much more stable over time than other symptoms. They may predate in undiagnosed state the first psychotic episode by years and also continue to be present after the remission. Negative symptoms include blunted affect, alogia, anhedonia, apathy, lack of motivation, and asociality. A long-term occurrence and lack of effective treatment causes them to have huge impact of patients' quality of life and their functioning in society. Therefore, there is a need for preclinical study to better understand the etiology of these symptoms and to identify possible treatment. Studies on negative symptoms in laboratory conditions because of their nature were limited. They were usually restricted to the social interaction test. And here the analysis of ultrasonic vocalization may help to fill this gap and complement the results obtained from the social interaction test and other tests. So although rat vocalization cannot be directly transmitted to human communication, the changes in vocalization may serve as a readout of negative symptoms. It was already said today, but I will quickly only remind for the clarity of the presentation that rat vocalization can be divided into three groups: for those emitted by pups and those emitted by juvenile and adult rats, and the latter vocalized with calls of mean frequency 22 kHz, which indicating on aversive state in rats, and those emitted and on calls of mean frequency 50 kHz that indicate on positive state. For the first 3 weeks of life, pups are entirely dependent on parental care, and every instance of separation from mother poses serious threat through their life. And it has been found that in such circumstances, pups vocalize with calls of mean frequency 40 kHz. And it's presumed that primary purpose of these calls is to induce maternal retrieval. But in addition to these communicative rules, they may also be a marker of pup distress. Neonatal vocalization are sensitive for many factors, including recent social stimuli. And based on the spectrogram and call characteristic can be classified into three general groups: short, flat, and frequency modulated sounds. Preclinical studies in the fields of schizophrenia require manipulation that will induce abnormalities in processes assessed. The models that are currently used for study in the studies of schizophrenia are drug-induced, neurodevelopmental, and the genetic. Pharmacological models are based on the administration of psychostimulant drugs, such as a dopamine enhancer or an NMDA receptor antagonist. intern neurodevelopmental model involve the environmental manipulations, diet or drug administration during the perinatal or early postnatal stage. And here we've got, for example, models that are based on neonatal lesion of right ventral hippocampus. Another strategy involved the inducing maternal immune activation, but by bacterial LPS or viral poly-IC, and studying the consequences of offspring. Schizophrenia phenotype can be also be induced by the compounds prenatal exposition to the methylazoxymethanol acetate, and I will talk a little bit more about this model later. And finally, we've got also genetic model, which are based on gene stability for schizophrenia. It's believed that neurodevelopmental models are the most valuable, as they mimic not only the symptomatology of disease, but also biochemical and neuroanatomical background of diseases. The knowledge of the vocalization in schizophrenia models are limited. The available reports were focused usually on the number of calls and demonstrate usually the depletion of vocalization. And for example, in pharmacological model, as early as 2000, Pups and Barkov demonstrate that administration of MK-801 reduces the number of 50 kilohertz sounds, and the similar effect has been observed also after the PCP administration and ketamine. And in the immunoactivation part, after the prenatal exposition to LPS, lower number of 40 kilohertz sounds have been found in isolated pups, and similar results have been found for poly-IC model. And in MAM model of schizophrenia, studies were focused only on the alert vocalization, and greater number of these sounds have been found in the juvenile rats. In our laboratory, we extended studies on poly-IC model in rats, and we found that poly-IC pups, when were separated from mother, they vocalized with reduced number of sounds, and this deficit was stronger in males, as they did not distinguish between social and unsocial stimuli. And poly-IC juvenile rats, when were played with partner, vocalized with reduced numbers of sound. And again, this effect was stronger in males. And this vocal disruption was associated with disruption in behavior. We also extended study on MAN model of schizophrenia. And in our hands, MAM juvenile rats, when well play with partner, they vocalize with a reduced number of sounds. And this deficit was also observed. We also observed the lower involvement of this animal in play with partner. These animals, when also vertical, they demonstrate tendency to vocalize with lower number of 50 kilohertz sounds. And the same pattern of changes we observe also in other rats during the social interaction. The poverty of the communication was not only observed in the number of emitted sounds, but also in the call complexity. And in both juvenile and adult MAM rats, during the social encounter, but also when animal were tickled, we observed the reduced bandwidth. And the lower complexity was also expressed by lower distribution of frequency modulated sounds. And this effect we observe in juvenile rats during the social play, but also when animals were tickled. And here we found the increasement in the number of alarm sounds that stay in line with data that I presented during the introduction. And a lower amount of frequency modulated sound have been also noted for the adult rats during the social interaction. I decided to further explore the vocalization in the MAM model of schizophrenia. This time I focus on the pup vocalization to obtain complex picture of vocalization in MAM animals. And the rationale of my study were limited data on ultrasonic communication in animal model of schizophrenia, especially during the earliest stage of animal life. I was also interested in the course of vocal development, as they are rappers on the occurrence of socio-emotional abnormalities prior to the onset of the psychosis in humans. And in my studies, I included both sexes, since the majority of data was done in males. And in accordance to NIH is to balance sex in preclinical studies. There are also sex differences in social-emotional functioning in humans. A few words maybe about the model that I use. It's evoked by the prenatal exposition of metalazoxymethanol. This is compound is genotoxin that selectively affects the neuroblast in certain nervous system, and the effect of this compound depends on the gestational day of administration, with 17 days of pregnancy being the most commonly used in the studies of schizophrenia. And administering MAM to pregnant dames results in neuroanatomical, electrophysiological, and behavioral changes in offspring, and these changes are similar to those observed in humans. Behavioral changes include positive-like symptoms such as hyperlocomotion or weaker prepuce inhibition. Cognitive impairments involve the weaker memory, cognitive inflexibility, and among negative-like symptoms, deficit in social interaction and ultrasonic vocalization has been found, as I mentioned before. I recorded Pups under 3 different conditions. I checked the sensitivity to social context, to social stimuli. Next, I assessed the course of their vocal development. On the final stage, I assessed how these pups are sensitive on the maternal potentiation effect. In my analysis, I take into account the number of sounds and call characteristics, such as a peak frequency, call bandwidth, and mean call duration. I also assess the call type distribution. Pup vocalization was induced by separation from mother, and they were recorded under 2 different conditions. In Y's case, they were placed on the bedding that was obtained from the home cage just before recording, so had familiar structure and smell, and it considered to be safer for pups. And in the second case, pups were placed on the novel bedding, which was more stressful, and we hypothesized that home cage bedding would have calming effect of pups vocalization. We expected to observe the increasement in the number of sounds in pups that were under more stressful conditions, but we couldn't catch this effect here. However, calls of these pups were much longer and had a lower frequency, what suggests that this pattern of changes is reserved for the pups that experience greater distress, and this effect was common for both control and MAM animals. We did not find the differences between number of emitted sounds in MAM animals, but the bandwidth of their calls, independently of the testing condition, was lower. Was lower. Also, distribution of call subtype changed, and in this model, and MAM pups vocalize with greater amount of flat sounds. To summarize this part of study, pups on novel bedding emit sounds of lower frequency and longer duration. MAM pups distinguish between present stimuli, and that suggests that these animals had unimpaired social information processing. MAM reduced the call complexity, what was expressed in the lower bandwidth and greater amount of flat sounds, and MAM effect was common for both sexes. In the next stage, I will talk about developmental changes in the vocalization in MAM model, and to this aim, I conducted the maternal sepulation test once again, and pups were recorded on 6, 9, and 12 postnatal day. Over the course of the time, in control animals, we observe the increasement in the number of sounds, and the skulls with the age of the animals become shorter. And we didn't observe this pattern of changes in mom models. Mom pups vocalize almost equally through the whole recording days. In control animals, we observe the increasement also in the call complexity and call frequency. And although this changes was not significant, but it appears that mom pups vocalize with the lower bandwidth and lower frequency. Older animals also vocalize with more complex sounds, and this effect was common for control and mom pups. With the age of control rats, the number of sounds grew, and this call becomes shorter with greater bandwidth and high frequency. Number of calls and their duration did not change in mom pups within the testing days. Mom pups demonstrate tendency to vocalize with calls of lower bandwidth at the lower frequency. And again, this effect was common for both sexes. One of the aspects of separation calls is maternal potentation effect. And this phenomenon can be defined as a increase in the rate of the sounds in re-isolated pups that were brief contact, that had brief contact with mother. And this phenomenon maybe is a model, maybe a model of the earliest life social bonds, especially mother-infant relationship. After initial isolation, pups were placed in a down holding cage, left there undisturbed, and during this reunion period, maternal behavior and pups' behavior were recorded, and then pups were re-isolated. We didn't find the differences between active care between mothers of mom and control pups. However, mom pups spent less time in direct contact with mother during the passive care. As we expected, we observed the increasement in the number of sounds during the re-isolation in control pups, and these calls were longer. We did not observe differences, this potentiation effect in respect to the number of calls in mom rats, although the re-isolated calls also in mom animals were longer. And independently of the testing condition, mom animals produce a lower number of sounds in comparison to control. Their calls were longer, had lower bandwidth, and lower peak frequency. And again, they vocalize with a greater amount of flat sounds. Re-isolated control pups, after brief union with the mother, vocalize with greater number of longer sounds, what we explain as a potentation effect. This potentation effect was not observed in the number in mom pups in respect to the number of sounds. Mom reduced the number of calls, and the calls were generally longer, less complex, and had lower frequency. And again, this effect was common for both sexes. The general conclusion is that prenatal exposition to metalazoxymethanol reduces vocalization in the offspring, and this reduction was expressed by the reduced number of calls, but also a lower call complexity, lower bandwidth, and greater distribution of flat sounds. Sometimes we observed that mom calls had a lower frequency and longer duration, and these changes indicate that mom animals may are more sensitive to stress factor. Together with the data that I presented in the introduction, it appears that that deficits which are evoked by prenatal exposition to mom are stable through the whole life of animals, but the degree of impairments depend on the day of animals and the stress factors. And this early communicative changes may be the predictor of changes in adulthood and may be relevant for negative symptoms. This study was supported by a grant obtained by Professor Agnieszka Potasiewicz, and I would like to thank all colleagues from my lab, especially Professor Nikiforov.